The overall goal of the following experiment is to show where a particular gene is expressed within an intact embryo. This is achieved by fixing the RNA to be localized in place within the embryo. As a second step, an antisense RNA probe specific to the gene of interest is hybridized to the target RNA within the embryo. This probe contains an epitope, usually digoxygenin, which can then be detected. Next, an antibody coupled to alkaline phosphatase is hybridized to the epitope on the RNA probe, followed by use of a colorimetric stain in order to visualize where the RNA is localized within the embryo. Results are obtained that show precisely where the gene of interest is expressed, based on the color reaction and knowledge of the embryo anatomy.
The main advantage of our in situ protocol over existing protocols is that it is faster and utilizes fewer reagents with no apparent loss of quality. This method can answer key questions in embryology, such as how tissues are specified and patterned.
Though this method can provide insight into the development of Xenopus, it can also be used for many other small embryos, such as zebrafish and early mouse embryos. We first started developing these methods because the original methods occupied a great deal of time, and we started experimenting with leaving out steps to increase the efficiency of the procedure without compromising quality.
Embryo fixation is performed in stages.